Thank you all. I'm very excited to now start with the talks. So what we put together is a series of talks of folks that were in the PRL in the 80s, 90s, 2000, 2010s. And they're going to talk to us about their experience at the PRL, about their science, and what the PRL meant to them and their career. So I'm really fortunate to introduce Mary Lou Guerino, who is at the Dartmouth College. Mary Lou, do you want to come over?
Okay, well, welcome to the 60th anniversary celebration. And what I'm going to talk to you about over the next 40 minutes is this talk I'm going to give you on it's elementary, making food more nutritious. And I'll explain how I captured this image in a little bit, but this is taken with the synchrotron imaging, and it's a leaf of Arabidopsis, obviously, my favorite organism. And this little ring at the base of each of these hairs is where zinc is stored. And then the blue is calcium, and then this is potassium found throughout the plant. So, it's not advancing. There we are. All right, are we ready?
Mm-hmm.
Okay, we're ready?
Yeah.
Or not.
Yeah, please, Tyler, go.
Come on. Okay. I realize not all of you will recognize this song immediately. Okay, I'm not playing the whole thing. It was the time of my life. Okay, we were ready for the 80s. And I think these lyrics, which you may not know by heart like I do, really summed up the 80s at the PRL. In the 80s, we will travel far, we will realize just who we are, and we can move as one. And this is a group of the 80s, the Hack and Go golf tournament. And I think it's from about 1985, because I recognize some of the grad students from our lab. We have Tom, we have Todd, here's Mark Estelle, Peter McCourt, Callie, all great times together. So we just heard from the Village People, getting us ready for the 80s. So here are the PRL people of the 80s. So, of course, Anton was the founding director. I was always terrified of him the whole time I was here. He was succeeded by Hans. I'll have a Hans story for you. And then this guy right here, Charlie Aronson, was the director when I came to the PRL. And one of the mandates for Charlie was to bring molecular biology to the already great plant science. at the PRL, and he hired these three guys. Barry Chelm, who I came to work with, I'll say another few words about that. Lee McIntosh, and of course, some of you may recognize Chris Somerville, who became a faculty member here about 6 months after I arrived. So this is less exciting look of the 80s at the PRL. And in case you're wondering where I am, whoops, this is not working again. I'm that one right there. So how did I arrive at the PRL? Well, I'd love to say it's because I knew its reputation and it was the greatest plant research group in the world and I had to be here, but that isn't what happened. What happened was this guy, my partner and now husband, Rob McClung, wanted to come to Michigan State to work with Frank Dazzo. And so I was finishing up a postdoc at Maryland, and I had to find a postdoc. And they had just hired Barry. In fact, he wasn't even here yet. And so he took a chance on me. And I'm telling you, he really took a chance because I had done a diving project for my PhD up in Halifax, Nova Scotia. And I always used to say, you know, I knew nothing about plants, so there's me, cute little diver on the boat. But of course, I did know something about plants because I was working on sea urchins and their diet. And these are my sea urchins lined up on a holdfast of kelp, chewing away. So that was food for my sea urchins. That was my plant background. And then I had done my first postdoc as an oceanographer in Rita Caldwell's lab, so I spent a lot of time going on cruises. But the one thing that did connect Barry Chelm, me, oceanography, and diving was nitrogen fixation. So Barry came here having worked on Bacillus phage, but he was a molecular biologist. And he decided he was going to work on the soybean rhizobia symbiosis. So can you imagine that today? You're a young assistant professor and you say, I'm going to work on something completely different. I've never worked on it before. But I had worked on nitrogen fixation in the sea urchin gut and then in the ocean waters, and I knew how to measure nitrogen fixation. So Barry's like, all right, what a team. I can clone genes, you know how to measure nitrogen fixation. So that's how it happened. So I became a nitrogen fixer. So at that time, here's me and Barry. And then there were 3 other labs, Frank's lab that Rob came to join, but ultimately he bailed and came to Barry's lab because the molecular biology was just so exciting at the time. Ken Nadler, and then Carl Schubert. So there were a group of us, and I like this quote from Frank because, of course, photosynthesis is high on the list here at the PRL these days. But nitrogen fixation is considered second only to photosynthesis as the most important biochemical reaction on Earth. So there was a whole group of us. It was very exciting times to be working on nitrogen fixation. And of course, I was working on soybeans, but it was also super exciting because of Arabidopsis. So it was really the beginning of molecular genetics everywhere, not just at the PRL. We were using Xerox pages from the yet-to-be-published Maniatis et al. So here's the cloning manual, which some of you more senior people may recognize. So we'd get these protocols from across the street. Ed Fritsch was in biochemistry. You know, and I made a Lambda library. I would do all these things just by following it like a recipe. And it worked. In fact, Peter Welk used to call me Magic Fingers because I could clone anything. And one of the grad students in the lab used to pay me. Well, we would reciprocate. He would take my sequencing gels apart because I hated ripping those glass plates and tearing my gel, and I would clone his genes. Okay, so Chris was there. He was telling everyone they had to work on Arabidopsis. And the excitement was really palpable, especially during the weekly seminars when we would have a lottery as to how many gel photos would be shown during that person's talk. And one week, Hans had 1 gel, and when the person showed the second gel, he leapt up, tore up his ticket. And of course, the speaker had no idea what the heck was going on. And after that, we were banned from guessing how many gels were going to be shown during the talks. And I know it's hard for you to see, but here's the 2 stars here, Chris and Lee. This is PBR 322, the vector. Puck 18, they're going to use some PST1 and some HEPA1. Here's the open vector, and they're going to pop this in. So they were planning their strategy in the old days for cloning. All right. So Hans loved a good practical joke. I put this picture in. There's me. And I'm actually, I think we're at Mark Estelle and Sylvie Dar's wedding. That's why we're all dressed up. But that was my interview suit when I went on the job market. And back in the day, there was no email and no text, so you get phone call. So I got a phone call from Elaine Tobin saying, oh, we'd like you to come out to UCLA and interview. I was so excited. It was going to be my first job interview. Hang up the phone. A few minutes later, Hans Kendi comes in. He goes, I hear you have a job interview at UCLA. And he's on the first floor. He never comes up to the third floor. I'm like, how did you know? And then I realized it was a joke. And that Hans had set me up, and it wasn't really Elaine Tobin. I was so mad. I was so mad. And he said, I would never do that to you. But I was convinced the whole day. Then he got mad at me for thinking it was him. So I had to call Elaine Tobin up. Say, hi, Elaine, I just wanted to check a few details. And it turned out I did an interview at UCLA. Okay, although they didn't offer me a job. But that was my favorite Hans Kendi joke. And some of the, well, again, more senior members may have other stories they'd like to share later about the jokes that he did play on people. And then the other thing I'll mention from the 80s was this very important meeting took place at MSU. And I had started my lab working on rhizobia and soybean at Dartmouth. But I came to the conference because in the back of my mind, I was always thinking I was going to work on Arabidopsis. And at this particular meeting, this is the Somerville lab, Trendies in Plant Genetics. They're all wearing the meeting t-shirt. But at that meeting, you know, they were really excited. There was tDNA-mediated transformation now of Arabidopsis. We were establishing community resources where you could write for seed and clone genes. And genetic mapping efforts were underway, which of course would ultimately lead to the sequencing of the first plant genome, that of Arabidopsis. Okay, so it took me the entire 80s to think about Arabidopsis. I finally started working on it in 1990. And so I'm going to tell you about not everything I did, but some of the things I did since I left the PRL that have to do with our work mostly on Arabidopsis. So many of you know I work on iron. Iron deficiency is the most common human nutritional disorder in the world today. And the reason for that is most people get their iron from eating plants. And iron is one of the 3 nutrients that most commonly limit plant growth. And unlike the other 2, nitrogen and phosphorus, you can't fertilize plants with iron because it precipitates out of solution at neutral pH in an aerobic environment. And so I have here a picture of iron deficiency anemia from WHO. And the darker the color, the worse the anemia. And so this is the main problem. People are largely eating plant material. They're not getting all of the important micronutrients they need. Here's some soybeans that are iron deficient, and here's some that are iron sufficient. And I also will mention this spinach is not a good source of iron, despite what your mother may have told you. Okay, because when they measured spinach, originally it had a lot of dirt in the crinkles, and most of the iron was in the soil and not in the spinach, but if you wash the spinach, then it's like any other leafy green. So for all of these that ate spinach for all those years, it's fine, but it wasn't giving you extra iron. Okay, so I had very simple questions to ask when I started my lab. Finally, moved to Arabidopsis, which I also did pre-tenure, which was also insane, but at the time, again, you just say, yeah, I'm switching, I'm not going to work on that anymore. Okay, so how do plants take up iron? How do they distribute iron? How do they store iron? And then how do they regulate iron homeostasis? Because all iron uptake is regulated in people, plants, microbes, and that's because iron can be quite toxic. So it's an essential element, but it can participate in Fenton chemistry and make hydroxyl radicals, and then those damage biomolecules. So all organisms are going to regulate its uptake. So plants like Arabidopsis, tomato, and soybean, they're going to do a couple of things when they sense, and we still don't know how they sense that they're iron-starved. They're going to start pumping out phenolics to help solubilize and chelate iron in the soil. And iron is the fourth most abundant element in the Earth's crust. It's just insoluble. It forms oxides and hydroxides. They're also going to pump out protons to help lower the pH. For every 1 unit drop in pH, iron becomes 1,000-fold more soluble. And then this solubilized iron 3 is going to be reduced by a membrane-bound reductase, which my lab identified many years ago in a Arabidopsis screen. It's going to take iron 3 to iron 2, and then the iron 2 is going to be transported back in via the IRT1 transporter, also identified in my lab. Rice, wheat, maize, and sorghum, the grasses do something a little different, and they act more like microbes. They're going to release small molecular weight compounds called mugenic acids. These actually chelate iron 3 directly, and then those iron 3 Ma complexes come in through a family of YSL-like transporters. But grasses can also bring in iron 2, if it's present, via IRT1-like transporter. So a good example would be rice growing in paddies where there's a lot of reduced iron. They can just take up the iron without it being chelated. So the first story I want to tell you is one about abundance. So we found a mutant with increased iron content in Arabidopsis. I call this my 3 graduate student project. Started with Maria. She passed it to Garo. He passed it to Nabila, who's just finishing it up now. And this came out of our work in what we call ionomics. So ionomics is a real thing. You can read about it in Wikipedia. But basically, it's an investigation of the genes involved in a fundamental feature of all living systems. That's the selective accumulation of ions. And so what we were doing was coupling genetics with elemental profiling. So we had different sources of genetic variation, EMS mutants, tDNA insertion lines of Arabidopsis, natural variation using accessions, and we also did extensive work on rice cultivars. So we could measure how much of an element was there using inductively coupled plasma mass spectroscopy. And I'll also talk about how we could localize elements using synchrotron X-ray fluorescence microprobe spectroscopy. So after 10 years of screening and 100,000 samples, we finally found this mutant. And at the time, the tech that was running all the samples called me up, because there was a group of us. We had a plant genome money, and one worked on salt, one worked on calcium, I was the iron maiden. And we had never found an iron one. And there, he says, do you want to work on this one? Like a joke. Okay, yes, I did. So here's wild type, about 50 parts per million in the shoot of iron. And here's our mutant called Brutus. It has an insane amount of iron in those leaves. And then we can also look at seeds, because of course seeds are what people eat. And we can grow the plants with normal level of iron, or we could give it high levels of iron. Well, wild type has homeostasis, so it doesn't matter how much iron you give it. It's controlling its uptake and partitioning, and it's about 40 parts per million. But you can see Brutus has already more than twice as much in the seed. And if you give that plant more iron, it just takes up more iron. In fact, it'll kill itself. And it turns out to have a constitutive iron uptake response. Now, it turned out when we were phenotyping this mutant, it grew better in alkaline soil, but as I already alluded to, it's very sensitive to iron. So here's our normal lab soil. Wild type looks fine. You can see Brutus is already a little small. If we make that soil alkaline by raising the pH to make iron less available, wild type's unhappy, but now Brutus is thriving. And if we add iron back to show it really is the lack of iron in the alkaline soil, wild type thrives and we kill Brutus. So why might that be cool? Well, 30% of the world's soils are alkaline. So I have a map here, and in blue are alkaline soil, and in red, acidic soils. And then here is pH increasing from 3 to 8, and percent availability of an element. And iron's here in the back, and you can see it really starts to plummet as the pH increases. So the idea then would be that we might be able to use plants that have a mutation in Brutus to grow in alkaline soils where other plants wouldn't be able to grow. So what is Brutus? Well, Brutus turns out to be a sensor in the iron regulatory cascade. So I'm going to spare you all the years of work we spent on this cascade. But just to run through quickly, there's a BHLH transcription factor. It's made all the time. We found that in our lab. And under iron deficiency, it gets phosphorylated. It then can form a heterodimer with there's 4 members of this group 4C BHLHs. That heterodimer comes over here, turns on another group of 4 BHLHs. They then go down here and form a heterodimer with FIT, another BHLH that we discovered over 20 years ago. And this heterodimer finally turns on the transporter, the reductase, the other machinery you need to take up iron. So what's Brutus doing? Well, Brutus is an E3 ligase, and it has 3 hemerythrin domains, and they can bind iron. And when this protein is bound to iron, that's its active form, so it's going to ubiquitinate substrates and target them for degradation. And so in the presence of iron, these transcription factors get turned over by Brutus, and then there's 2 other Brutus-like proteins, and they turn over this group of heterodimers. So when iron's around, the transcription factors get turned over. When iron isn't around, Brutus is inactive, and you allow transcription to occur. Now, annoyingly, the same genes were discovered using, of course, the information from Arabidopsis, but they changed the name. But HRZ in wheat and in rice is Brutus, and these lines that have mutations also accumulate iron, and they grow better under alkaline conditions. So these stories are just coming out. There's one bioRxiv paper online right now about wheat, but I think it's a sort of a nice story of how starting in Arabidopsis, and then it does translate. And of course, the loss of a gene is a lot easier than trying to mess up and change function. Just get rid of it, they grow better. There's just too much negative regulation in that pathway. And that's what this one is trying to show you here. So just the darker the color, the more iron. We can eliminate Brutus 1-like gene, Brutus-like 2. This is an allele of Brutus. It's a little milder than ours. And by the time you get rid of all 3, a lot of iron in the leaf, the plants look great, and they have high chlorophyll, compared to the wild type grown under minus iron conditions. So you can ask yourself, because I've been asking myself for many years, why you need over 20 transcription factors, 3 E3 ligases, a kinase, and a few other things to just tell the plant whether it should take up iron. Okay, story number 2 is about localization. And we discovered that there are really different patterns for different elements. And so this has been worked on over the years by a number of people in the lab, both postdocs and students. The technology was actually brought to my lab by a postdoc, Tracy Punchin. And this is a seed story. We were down in the roots for so long, we had to skip the leaves and just go right to the seeds. We wanted to know, since seeds are a major food source for most of the world, how are the nutrients getting into the seed? Well, it turns out we still don't really understand how nutrients end up in the seed. That's for almost any nutrient. And we know that consuming seeds with low micronutrient content is the problem that causes these micronutrient deficiencies. In addition, if you had nutrient-rich seed, you get agronomic benefits such as increased seedling vigor, resistance to disease, and of course, ultimately increase crop yields. So we use synchrotron X-ray fluorescence microprobe spectroscopy. So some of you may be familiar with the use of the synchrotron to do crystal structures of proteins. But what we're doing is we're accelerating electrons at close to the speed of light. And as they are circling in the synchrotron, the path is being bent by strong magnets. And every time the path gets bent, energy is given off as X-rays. They have different characteristics. And so then these X-rays go to these beam lines, and you're either getting your crystal structure of your protein, or you're figuring out where your elements are in your Arabidopsis seed. Because as we raster along a sample, at each point that we point the X-ray, we collect a full spectrum of ions, and that's because each atom gets excited by the X-ray, and then it's going to fluoresce with a particular signature. So we can tell iron from manganese, from zinc, from copper, etc. And then you take all this information. Well, I don't. The physicists take all this information and make an image that I can understand. My role is to say that doesn't make any sense, and then they go back and do it again. So this is when this has got messed up. When we first started out, so here's the seed, the radicle, the hypocotyl, and the leaves, the embryonic cotyledons. This is the level we were imaging. Wasn't great. But this is where we are now. So this was about a 10 micron resolution. This is under a micron. Now you can see in green there's some stripes of manganese. There is the cells that surround the vasculature are storing iron, shown in red. And if this slide had the right background, which is supposed to be black, this is a 2D image, right? We're just shooting directly at the seed. But we can also look at the seed using tomography. And when we do tomography, then we can get, it's like confocal, you get sections. So now what you're looking at is a section of about here in our Arabidopsis seed. And you can see and count actually the cells that are storing iron. Those are the endodermal cells. And then you can see there's a single layer of cells that are storing manganese. And then you can see these are the veins in the cotyledons, and they're also storing iron. Okay, so that was pretty interesting. So these unique patterns raise a lot of questions, right? How and when are the patterns set up? Do they matter to the plant? And what happens when the seed germinates? So I'll show you we've answered these two questions for some of the elements. This one is a technical challenge that we haven't cracked yet. But this whole story, this using the synchrotron, came out of asking this question, one of my 4 questions. Where do seeds store iron? So our hypothesis was that plants were going to store iron in their vacuole, just like yeast. And then our second hypothesis was if plants can't put their iron in the vacuole, they're going to have less iron in their seed. So the strategy then would be to increase the iron content by putting more iron in the vacuole. So we were right about the first one. Plants do store iron in the vacuole, but we were not right about the second one. And we discovered this when we made a mutant of the vacuolar iron transporter. So we were lucky there's only one transporter that moves iron into the vacuole. We called it VIT1. And down here is the amount of iron in the seed of a wild type or the mutant. And so this is where we were before we started imaging. We knew it had exactly the same amount. So the postdoc that was working on it at the time is like, well, that was a waste. You know, that isn't going to change the iron content of the seed. But I had the new postdoc, Tracy, come in, and she knew how to do the imaging. So these are some of our first images at the lower resolution. And we had the 6 blue donuts. But you can see that that VIT1 mutant was putting iron in an entirely different place. And then we asked, did it matter? And it did. So here's wild type and VIT1 at our sort of normal soil pH. raise that pH to make iron less available. Wild type's unhappy and chlorotic. We kill VIT1. So VIT1 germinates and dies. So our interpretation is that you need to be storing iron in your cells that surround the vasculature, because that's the iron the plant's going to use first as it germinates before the root comes out and is able to take over. And at the time we were working on iron into the vacuole, a group in France was working how iron gets out of the vacuole, and he got the same result. So if you can't get it out of the vacuole, you also die. Okay, and then that's the really pretty picture that we love to show, those cells, the storing iron. And then the manganese, I think that's next, yeah. I just put this in case anyone's red-green colorblind, because everything coming out of the synchrotron was red, green, blue, and my collaborator, David Salt, is colorblind. And he'd always be like, oh, that's beautiful. Yeah, brown. Okay, so it took us a while to get around to looking at the manganese stripes, but there was a family of metal transporters called the MTPs, and there were 11 of them, and there were 4 that were predicted to transport manganese. So we did a quick 2D synchrotron imaging of the mutants, and you can see the stripes now. Manganese is sort of in this reddy purple. MTP 9, 10, and 11, the stripes are still there. MTP 8, now the manganese is where the iron is, iron shown in blue. Okay, so we have not answered the question of why the manganese is stored there, so when those cotyledons open, that manganese would be on the underside of the leaf. Of course, manganese is very important for the water splitting reaction in photosynthesis, but why you would store it there, I have no idea. Okay, so once we knew it was MTP8, then we can do the higher resolution work at APS. I should mention all the synchrotrons are managed and funded by DOE. So anyways, a little commercial for DOE. So now here is manganese, and in the VIT1 mutant, same as wild type, but the MTP8 mutant, the manganese now where the iron is. I had shown you the iron pattern where now the iron goes to where the manganese is. And if you get rid of both, the patterning disappears. So this was very satisfying because it meant these 2 genes set up all the patterning for iron and zinc, iron and manganese. Okay, moving forward then, we gotta translate our work. So cassava is a very productive, resilient staple crop, grown largely in sub-Saharan regions of the world. It's consumed by 800 million people daily, and it's a very important source of dietary calories, because that's all you're getting is calories, no other nutrients. So here's some cassava, here's the starchy root, and this is a map of cassava production. So it turned out that there had been a large effort by the Gates Foundation, funded by the Gates Foundation, to improve cassava's nutrient content, both for micronutrients, et cetera, and vitamins. And this work was being done at the Danforth Center, and for about 10 years, they had a strategy for increasing the iron content, and let's just say it didn't work. So at that point, Debbie Delmer, another PRL alum from the early days, she told them, the Gates people, they should ask me. I was like, okay, so they flew me to St. Louis, and they asked me, what do you think we should do about this cassava? So I said, well, you should express FIT1 in the starchy root, and then it will accumulate iron, and maybe increase the transport, express IRT1. But I also said, I know nothing about cassava. I know nothing about how starchy roots take in iron, so you should give me some money so I could study that. And they told me, no, we have to be in the field in 5 years. We're doing translational research. The government pays for the basic research. Okay, well, they took my advice. They put, I never got a penny from Gates. They put VIT1 into cassava, expressing it from a promoter that's only expressed in the starchy roots. It increased the iron content in the greenhouse. They did field trials in Puerto Rico. It increased the iron content in the field-grown cassava. And then this paper came out in 2019, And I was super proud of it, even though I'm not an author on the paper if you're busy reading, because this cassava that was overexpressing the Arabidopsis VIT1 transporter had 3 to 7 times higher levels of iron in those storage roots. So that would be about 50% of your estimated daily requirement if you were eating cassava as your main staple. And then plants that were overexpressing the iron transporter and ferritin, which is a cage that you can put a lot of iron into as a storage form, they had even higher levels of iron. And IRT1, I didn't tell you, also transports zinc. So now you're getting higher zinc. And now you're up to almost 70% of your daily estimated requirement for iron. And I did get thanked. OK, there it is. For performing an advisory role, which I thought was a little light on the thanks. But it's OK. It's OK. I'm very proud of the work. I know we have a Danforth person sitting right there. She's probably going to report back everything I said. OK, then my third story, which is abundance and location, is on arsenic in rice. And this was done in collaboration with rice breeder Shannon Pinson and ionomics guru and my longtime collaborator David Salt. And they're both retired, which is probably telling me something, like maybe I should too. OK, so I wanted to talk about arsenic because it raised its little head again. July 1st, 2025, this article came out in the New York Times. Should I be worried about arsenic in rice? The answer is yes. OK, and that was because this study had just come out from the Healthy Babies, Bright Futures, and they were looking at a variety of toxic elements, lead, cadmium, and arsenic. And of course, they were finding it, and so it raised the alarm bells. However, back in 2014, there was another article in the New York Times. And this one was called The Trouble with Rice. And I'll just read you the first sentence. For the past few years, Mary Lou Garneau has been keeping watch over experimental fields in southeast Texas, monitoring rice plants as they suck metals and other troublesome elements from the soil. So when this came out, then ABC News called. They want to get a picture of me in the field. And I'm like, I'm in New Hampshire. I've never been in southeast Texas. So I was like, you have to call my collaborator, Shannon, because Shannon's the one that's in the field, every day with the temperatures over 100 degrees. But she was a little peeved that I got mentioned, but not her. OK, so but I had to make it up to her for a lot of years after that. But anyway, she was a great collaborator. And we did find there was a problem. So what's the importance of the problem? Arsenic is a class 1 non-threshold carcinogen. That means there's no safe level. And it's being found in rice. And rice is eaten by half the world every day. And many rice-based foods are on the market, including infant rice cereals and formula with organic brown rice syrup. So this is really key. This sounds great. They were using it to replace high fructose corn syrup. It's organic. It's brown rice. It sounds like just the thing you should be feeding your babies. And those of you who eat energy bars, like Cliff bars and things, if you look, it has organic brown rice syrup. But it turns out that's where the arsenic is going to accumulate, as we discover, is in the bran of the rice. And then, of course, people with allergies to gluten are consuming a lot of rice-based products. So arsenic occurs naturally in soil and groundwater. However, was also used for many years in pesticides and wood preservatives. And so what is it about rice? Well, there's several factors that contribute to rice being an efficient accumulator relative to our other cereals. First of all, as you know, we cultivate rice in flooded paddies, and that leads to mobilization of arsenic-3, arsenite. And then arsenite is inadvertently taken up through the silicon transport pathway. So this pathway, this transporter, can't tell the difference between arsenite and silicon. And this pathway is highly efficient in rice. Rice takes up 10 times more silicon than other grasses. And it uses it to strengthen its stem, and it has anti-herbivory properties. So here it is taking up all this arsenite, which is freely available in the flooded paddies. And then it also can't tell the difference between arsenic 5, arsenate, and phosphate. And so it's busy taking up arsenate. And so this is just some examples of arsenic levels in flooded rice versus unflooded rice. And these were just two of the parents for a recombinant inbred population that Shannon had made that we were using to do QTL analysis. And the reason, of course, is because we're getting this reduction. And then when it's reduced, it desorbs from the surfaces like iron hydroxides. It goes into solution. So that's how we get the arsenite. However, you might say, well, then let's not grow flooded rice. There is upland rice. And also to save water, people have talked about alternating wet and dry cycles for rice growth. But this is cadmium. So cadmium's increased whenever the plants are grown under unflooded conditions, because the cadmium sulfides would have precipitated out of solution in the flooded paddy, but now they're mobile. And so you really can't win with rice. You can either have arsenic rice or cadmium rice. Okay, so what are we going to do about it? Well, we need rice cultivars that restrict arsenic accumulation in the grain. We're never going to get rid of arsenic in the soil and water, so we just need to figure out how to prevent it to getting into the parts we eat. And if we did that, of course, we could use them immediately as genetic stocks in breeding programs and hopefully solve the problem. Of course, I'm an Arabidopsis person, so while Shannon is out in the field doing the hard QTL analysis, we're in the lab with Arabidopsis. So we did a GWAS, used 349 Arabidopsis accessions, and we did find a SNP that explained 18% of the total variation. This thing is not working every time. Right there. We called this gene HAC1, and what is it? It's an arsenate reductase. So this mutant that can't reduce arsenate to arsenite has 12 times more arsenic in its roots and 300 times more arsenic in its shoots because it can't efflux arsenic back out of the root. And I'll show you a little model of that as one of my final slides. But I will say that, once again, similar genes are found in rice and shown to function similarly. So what do we think is going on? Well, here comes the arsenate. Comes in as arsenic 5. It gets reduced by HAC1 in the epidermis, and then this can be effluxed, the arsenite, right back out. So that's a very important mechanism to prevent accumulation. And then if arsenate makes its way through the root layers and gets to the pericycle, then HAC1 is also expressed in this layer of cells. And now this arsenite is going to get sent to the vacuole. This may also happen in the endodermis. And it's conjugated to phytochelatins. And so then it's not mobile, and it won't move into the xylem for transport to the shoot. We've also done some imaging. And I'll just show you one example. We can pulse the grain with different forms of arsenic via the stem. And then here's the cross-section of the grain. And we discovered that arsenite's not very mobile. So it's just sitting up here. The OVT is the strand of vascular tissue in rice where the nutrients are going to come from the mother plant and go into the endosperm during grain filling. Okay, so they're going to move. But of course, they don't move, which is why rice is not full of great nutrients. It's also not so full of arsenite. But if we look at a methylated form of arsenic, DMA, you can see that completely fills the endosperm over that short period that we were pulsing. Now, plants don't make dimethyl arsenate, but microbes make a lot of it in the soil, and then plants can take it up. It's not thought to be as dangerous, but it just goes to show you that we really need to know what's going on with all the different forms. And with that, that's the end of my third little story. That one doesn't have a happy ending yet, but we're still working on it. Although with Shannon and David retired, I'm going to have to find some new collaborators. This is the group over the years that have worked in my lab, some of my great collaborators. And I have to thank NSF, NIH, DOE, Superfund Research Program, and NIEHS for funding these projects over the years. Thank you for your attention, and I'd be happy to take any questions.
Thank you so much, Mary Lou. Such a great talk. Any questions for Mary Lou? Actually, while they think about it, I have
Okay.
a burning question that relates a bit to Brutus, but maybe it's more general. So when you start modifying access of iron to the plant, what would happen to other nutrients? And I know very little about all these things, but if I don't go wrong, phosphate, uptake of phosphate follows a burst in iron uptake. So when you start changing these dynamics with ions, what would happen to other ions that sometimes
You
are
must
not
talk to
considered?
Hatim.
I had to talk. Well, we
Yes.
work on that. That's why I know. The little that I know is because of that.
No, he's here at Michigan State, and he's one of the few people that started to look at more than one element at a time. You know, like there's a whole group of people who work on phosphorus. I work on iron, others on zinc. So it does change the dynamics.
Right.
But, you know, again, now we need to go back and look more carefully. But certainly, you know, I was encouraged by the wheat and the rice results with Brutus that when you knock it out, the plants are doing better.
Right. It's fascinating, but I was wondering whether altering, the dynamics for other elements could be helpful for your arsenate or
Yeah.
for our arsenate problem. So,
Yeah, thank you. That's definitely.
Question from Sue?
Yeah, that was really exciting. I have a question about the iron localization in the seed that looks like these rings. Is that bundle sheath cells?
Yeah, bundle sheath in the cotyledons.
So what is it doing in the bundle sheath? Why that cell type?
Well, the only thing I can say, and this is where my lack of plant training will come in now, is they're surrounding the vascular cells. And so there must be a pathway out of bundle sheath into the phloem for distribution.
Yeah, I think one interesting possibility is that it's surrounded by suberin, so it may not be able to get out. So I wonder if there's something to that. There are suberin-less mutants out there too. But anyway, I'd love to talk more about that.
Okay.
Wonderful. Well, maybe in the interest of time, we have to move to
I
the
was
next-
on time, though.
No, you were perfect.
Okay. I just like to point that out. Thank
Okay.
you.